In this video, let us know about phylum Protozoa. Protozoan means the first animal. Protos means first. Zoan or zoan means animal. The name protozoan was first coined and used by George Goldfuss in 1818, representing the lower groups of zoophyta. But C.T. von Siebold recognized the unicellular nature and first used and defined the name protozoa in the present sense. Protozoans are eukaryotic, unicellular, and lack cell wall. Protozoans are simplest and most primitive of all animals. General characteristics of phylum Protozoa. Protozoans are small, microscopic in nature. They exhibit two forms of living. They are either free-living or parasitic. When they are parasitic, they are either ectoparasite, meaning they live on the surface of the host, or they are endoparasites, where the organism inhabits inside part of the body of the host. They exhibit protoplasmic grade of organization. The presence of one or more nuclei indicates the nuclear monomorphism or dimorphism. They are solitary or colonial. Further, the size of protozoans measures from 1 micrometer to several millimeters. The shape may be either spherical, oval, elongated, or flattened. They exhibit bilateral symmetry, radial symmetry, spherical symmetry, or none, which means asymmetrical. Their body form remains constant, which means in their period of growth, the body form do not change. The body protoplasm consists of ectoplasm and endoplasm. The locomotory organs are pseudopodia, cilia, or flagella. The body is naked or bounded by pellicle. Protozoans have various modes of nutrition. They are holozoic, holophytic, saprozoic, or parasitic. The digestion takes place intracellularly. Respiratory mechanism takes place by the process of diffusion through the body surface. Excretion is through general body surface or by cytopyge. The contractile vacuoles helps in osmoregulation. The reproduction is either sexual or asexual. The life cycle of protozoans exhibit alteration of generation. Classification of phylum Protozoa. Phylum Protozoa is further classified into subphylum, classes, and orders. Phylum Protozoa consists of 5 subphylums. They are Sarcomastigophora, Sporozoa, Nidospora, and Ciliophora. Subphylum Sarcomastigophora is subdivided into 5 classes. They are Phytomastigophoriae, Rhizopodiae, Zoomastigophoriae, Pyroplasmiae, Actinopodiae. The class Phytomastigophoriae is subdivided into orders. Order Chrysomonadina, Coccolithophorida, Heterochlorida, Euglinida, Volvocida, Dinoflagellida, Cryptomonadida. Next, class Rhizopodiae is subdivided into order Amoebida, order Arselinida, order Foraminiferida, order Diplomonadida. Class Zoomastigophoriae is subdivided into order Choanoflagellida, Rhizomastagida, Hypermastagida, Diplomonadida, Kinetoplastida, Bicosoicida, Retortomonadina, Oxymonadina, Trichomonadina. Subphylum Sporozoa is classified into three classes. They are Telosporia, Toxoplasmia, Haplosporia. Class Telosporiae is classified into order Eucoicida. Subphylum Nidospora is divided into class Myxosporidae and class Microsporidae. Subphylum Ciliophora is classified into class Ciliatae. The class Ciliatae is subdivided into 15 orders. They are Gymnostomatida, Trichostomatida, Conotrichida, Apostomatida, Astomatida, Hymenostomatida, Thigmotrichida, Peritrichida, Suctorida, Heterotrichida, Oligotrichida, Tintinida, Entodynomorphida, Odontostomatida, Hypotrichida. Subphylum Sarcomastigophora. The organisms of this subphylum consist of pseudopodia or flagella or both as their locomotory organelle. The nuclei is of one kind, which means monomorphic. The spore formation process is absent in this subphylum. The reproduction takes place by syngamy. Examples of Sarcomastigophora are amoeba, euglena. Subphylum Nidospora. The organisms of this subphylum are parasitic and lacks locomotory structures. The organisms have spore formation with one or more polar filaments. These spores are discharged and attached to the host during infection. Example of subphylum Nidospora is Myxobolus. Subphylum Ciliophora or Ciliates. The organisms under the subphylum Ciliophora are large protozoans. They might reach about 2 mm in length. Their locomotion is by cilia. The best example for subphylum Ciliophora is Paramecium caudatum. Subphylum Sporozoa or Sporozoans. The organism under this subphylum lacks locomotory organs because of the tendency to form spores. The gametes of subphylum Sporozoa has either cilia or flagella for their locomotion. The organism belonging to this subphylum are parasitic. The spore formation is the common function under this phylum. Example, Plasmodium. The examples of phylum Protozoa are Euglena, Amoeba, Plasmodium, Paramecium, Podophyra, etc. Under the phylum Protozoa, let us now study about Paramecium caudatum as the type study organism. Scientific classification of Paramecium caudatum. The organism belongs to kingdom Animalia, phylum Protozoa, class Ciliata, order Hymenostomatida, suborder Peniculina, genus Paramecium, species Paramecium caudatum. Habit and habitat of Paramecium caudatum. Paramecium caudatum is found in freshwater ponds, pools, rivers, etc. Paramecium caudatum is especially found in stagnant ponds, organic infusions, and sewage waters. They are free-living organisms. Appearance and physical characteristics of Paramecium caudatum. The organism measures about 170 to 330 micrometers long but are usually 200 to 300 micrometers long. They are spindle-shaped, which has rounded front and tapering posterior blunt point. As a whole, the organism shape is like a slipper and hence the name slipper animalcule. The organism is either grey or white in color. Paramecium are covered with minute hair-like organelles called cilia, which is used for locomotion and feeding. The anterior part of paramecium is slender with blunt or rounded end. The posterior part of paramecium is pointed or cone-shaped. The widest part is just below the middle. The pellicle is covered with cilia. The organism has a long oral groove. The oral cavity of the paramecium is lined with cilia. Paramecium chordatum has two contractile vacuoles which serve to excrete excess water taken up from the outside, thereby regulating the water contents of the body. Radially distributed collecting canals give the contractile vacuoles a distinctive star-like shape. The cell is enclosed by a cellular envelope which is called as the cortex, that is densely studded with spindle-shaped extrusomes called trichocysts. Paramecium chordatum feed on bacteria and small eukaryotic cells such as yeast and flagellate algae. The accumulated food particles at the posterior end of the cytopharynx are directed by the long cilia into the rounded ball-like mass in the endoplasm that is called as the food vacuole. The food vacuoles are circulated by the streaming movement of the endoplasm, which is called cyclosis. In hypotonic condition, that is in fresh water, the cell absorbs water by osmosis. It regulates osmotic pressure with the help of bladder-like contractile vacuoles, gathering internal water through its star-shaped radial canals and expelling the excess through the plasma membrane. When moving through the water, they follow a spiral path while rotating on the long axis. Pellicle. The body is covered by a thin, double-layered, elastic and firm pellicle which is made of gelatin. The pellicle holds the shape of animal but is elastic enough to permit contractions. The pellicle has double membrane. The outer membrane is continuous with the cilia and the inner membrane is with ectoplasm. Under the higher magnification of microscope, pellicle shows rectangular or hexagonal depression on its surface. This arrangement is preserved on the dorsal surface of paramecium, but on the ventral surface the ridges converge in front and behind towards a preoral and postoral aperture. Each hexagonal depression is perforated by a central aperture through which a single cilium emerges out. The anterior and posterior margins of hexagonal depressions bear the opening of trichocysts. The electron microscopic study of pellicle by Ehret and Powers in 1957 has revealed that the hexagonal depression correspond to regular series of cavities, the alveoli. All alveoli collectively form a continuous alveolar layer, which is delimited by an outer alveolar and inner alveolar membranes. The outer layer lies in close contact beneath the outer cell membrane. Therefore, pellicle includes outer cell membrane, outer alveolar membrane, and inner alveolar membrane. Cilia. The entire body is covered with numerous small hair-like projections called cilia. Cilia occur in longitudinal rows all over the body. This condition is known as holotrichus, in which the body cilia are equal. Cilia have the same structure as flagella. They have an outer protoplasmic sheath or plasma membrane with 9 double longitudinal fibrils in a peripheral ring. In some cilia, the 9 outer fibrils are not paired. There are 2 central longitudinal fibrils which are thinner than our outer fibrils. Each cilium arises from a basal granule or kinetosome. The 9 pairs of peripheral fibrils fuse together to form the wall of the kinetosome. Thus, kinetosome is a tube which is either open or closed at its lower end. The 2 central fibrils stop at the level of the pellicle in most ciliates. Arising from the kinetosome is a thin rhizoplast which does not join the nucleus. Many metazoa also have cilia. Their structure is the same, except that the basal granule is different and it has fine filaments or rooting fibers extending down into the cytoplasm. But cilia differ from flagella in being generally more numerous and shorter in size. The ciliature may be conveniently divided into body or somatic cilia, which are found on the body surface, and into oral ciliature, which is associated with the mouth region. The body cilia are equal, but they are longer at the posterior end, hence the name chordatum. The cilia are organelles of locomotion and food collection. They also act as sensory receptor and detect stimuli on the external environment. Ultrastructure of cilia. The cilia and flagella have a fibrillar composition. At the base, the cilium has the diameter of about 0.2 micron or 2000 angstrom, which may be up to 10 microns above the cell surface. The cilia are bounded by a unit membrane of 90 angstrom thickness, which resembles and remains continuous with plasma membrane. The bounded space of the cilium contains a watery substance known as the matrix. In the matrix, there remain embedded 11 longitudinal fibrils or microtubules. Out of the 11 fibrillars, 2 are located in the center, while the remaining 9 fibrils remain arranged peripherally around the central fibrils. Each of the 9 outer fibrils is 360 angstrom in diameter and composed of 2 subunits of 180 to 250 angstrom diameter. The infra-ciliary system is located just below the pellicular alveoli. It consists of kinetosome or basal body and kinetodesma. The cilia arise from kinetosomes, and from each kinetosome arises a delicate cytoplasmic fibril called kinetodesma. A longitudinal row of kinetosomes with their kinetodesmata forms a longitudinal unit called a kinete. The infra-ciliary system controls and coordinates the movement of cilia, and it brings about formation of organelles in cell division. Example, some kinetia from the mouth. In binary fission of ciliates, the kinetia are cut transversely into 2, each going to one daughter cell, that is called as perikinetal fission. Oral groove and cytopyge. On the ventrolateral side, there is a large, oblique, shallow depression called oral groove or peristome, which gives the animal an asymmetrical appearance. The oral groove leads into a short conical funnel-shaped depression called vestibule. The vestibule leads directly into the fixed oval-shaped opening called cytostome, which is the mouth. Extending directly from the cytostome Toward the center of the body is the white cytopharynx. The cytopharynx then turns sharply towards the posterior side to become the slender, tapering esophagus. Thus, the esophagus is roughly parallel to the body surface of paramecium. Here, the esophagus turns again toward the center of the animal to lead into the forming food vacuole. The cytopyge, which is also termed as the cell anus or anal spot or cytoproct, lies on the ventral surface of the body almost vertically behind the cytostome or mouth. Undigested food particles are eliminated through the cytopyge. The ciliation of cytopharynx is very complicated. Cytoplasm. The cytoplasm is differentiated into a narrow external or cortical zone called the ectoplasm and a larger internal or medullary region called endoplasm. Ectoplasm. The ectoplasm, which is also called as ectosarc or cortex, is a permanent part of the body, strikingly delimited from the endoplasm. Ectoplasm forms a firm, clear, thin, and dense outer layer. It contains the trichocysts, cilia, and fibrillar structures, and is bounded externally by a covering called pellicle. Endoplasm. Endoplasm is fluid, voluminous part which consists of all cytoplasmic inclusions. The inclusions are mitochondria, Golgi apparatus, vacuole, crystals, granules, chromidia, etc. Trichocysts. Embedded in the ectoplasm are right angles to the surface or small spindle-shaped bags called trichocysts. A small spot on each anterior and posterior margin of hexagon marks the position of trichocyst. They are filled with a refractive, dense fluid having a swelling substance at the outer end is a conical head or spike. The trichocysts lie perpendicular in the ectoplasm. They open by small pores on the ridges of hexagonal areas of the pellicle. They arise from kinetosomes of cilia, then migrate and locate themselves at equal distance in the endoplasm. When the animal is irritated, the trichocysts are discharged as long sticky threads. A discharged trichocyst has an opaque spike like an inverted nail and a long striated shaft, but the shaft is not seen in the undischarged state and is probably formed during discharge. The function of trichocysts is uncertain, but they are discharged as a reaction to local contacts and injury. They may serve as organelles of defense. Nuclei. In the endoplasm, near the cytostome are two nuclei, that is, paramecium is heterokaryotic, a large ellipsoidal and granular macronucleus, and other small compact micronucleus is present. Macronucleus is a conspicuous ellipsoidal or kidney-shaped body. It is of a compact type containing fine threads and tightly packed discrete chromatin granules of variable size and embedded in an achromatic matrix. It possesses many nucleoli and much more chromatin material, that is DNA. It is a somatic or vegetative nucleus. It divides amitotically and controls the vegetative function, which means the metabolic functions of the animal. It does undergo mitosis. Micronucleus is small, compact, and spherical. It is generally found close to the macronucleus. Fine chromatin granules and threads are uniformly distributed throughout the structure. The micronucleus divides mitotically and controls the reproduction. The number of micronucleus varies with the species. It is 1 in Paramecium caudatum, 2 in Paramecium aurelia, and many in Paramecium multimicronucleatum. Contractile vacuoles. There are 2 large filled contractile vacuoles, each situated near one end of the body close to the dorsal surface. Their position is fixed. They lie between the ectoplasm and endoplasm, but they are temporary organelles disappearing periodically. In some species, they appear to have a lining membrane, in which case they do not disappear entirely during systole. Connected to each contractile vacuole are 5 to 12 tubular radiating canals, each consisting of a terminal part, long ampulla which collapses when empty, and a short injector canal which opens into the vacuole. The canals communicate with large part of the body, from where they take up liquids and pour them into the vacuole, which is thus reconstituted and grows in size. When the contractile vacuole reaches its maximum size, it contracts suddenly, that is, the systole phase, where it contracts and discharges its content through a permanent pore in the pellicle. Then the canals again form the contractile vacuoles. The canals do not disappear entirely since they are permanent structures. The main function of the canals and the contractile vacuoles are hydrostatic. They remove excess of water from protoplasm. The water is partly absorbed and partly taken in while feeding. The nitrogenous waste substance such as ammonia and some urates are also expelled through the contractile vacuoles along with the carbon dioxide. Food vacuoles. These are roughly spherical, non-contractile bodies varying in size and number, lying in the endoplasm. They contain ingested food particles, principally bacteria, and a small amount of fluid bounded by a thin definite membrane. Volkonsky in 1934 proposed the name gastrioles for these vacuoles. Associated with the food vacuoles are the digestive granules. Locomotion of Paramecium caudatum. Paramecium caudatum performs locomotion by 2 methods, via metaboly or body contortions and by ciliary locomotions. Metaboly or body contortions. The body of Paramecium caudatum possesses elasticity. It can squeeze itself through a passage narrower than its body, after which the body assumes its normal shape. This temporary change of body shape is metaboly. It is brought about in Paramecium by the protoplasm. Ciliary locomotion. Locomotion brought about by cilia is the main method of locomotion. The cilia can beat forward or backward, enabling the animal to swim anteriorly or posteriorly. Normally, the animal swims forwards by the cilia beating backwards. At that point of time, the cilia becomes stiff and bends backward and almost touches the body's surface. This is called the effective stroke, which is very fast. Then, after the fast effective stroke, the cilia become limp and returns slowly to the original vertical position. This is called the recovery stroke. The coordinated movement of cilia is called as the metachronal rhythm. This is due to the infra-ciliary system. This causes swimming forward by the animal. Nutrition in Paramecium chordatum. In Paramecium chordatum, the nutrition is holozoic, which means the organism engulfs the food. The food comprises chiefly bacteria and minute protozoa. Paramecium does not wait for the food, but it hunts for the food actively. It is claimed that Paramecium chordatum shows a choice in the selection of its food. Feeding mechanism of Paramecium chordatum. When Paramecium chordatum enters a region of abundant food, it comes to rest. Paramecium chordatum feeds only at rest or when swimming very slowly. It never feeds when swimming fast. The beating of cilia of the oral groove causes a cone-shaped vortex of food-laden water to be swept into the oral groove from a distance in advance to the anterior end. The particles of food then go to the vestibule, from where some food particles are rejected and thrown out, but others pass into the cytostome. At the end of the cytopharynx, a food vacuole is formed and filled with particles of food. When the food vacuole reaches a certain size, the food vacuole is pinched off and started its course in the paramecium. The food vacuole contains some water besides the food. Digestion and egestion of Paramecium chordatum. Digestion occurs by enzymes secreted by protoplasm into the food vacuoles. In digestion, proteins are changed into amino acids, carbohydrates are changed into soluble sugars, and glycogen and fats are probably also digested. The contents of food vacuoles are first acidic and then become alkaline. The undigested matter is egested through the cytopyge with some force. Respiration and excretion of Paramecium chordatum. The exchange of gases takes place through the semi-permeable pellicle, like other freshwater protozoans, by the process of diffusion. Osmoregulation in Paramecium chordatum. Paramecium chordatum has two contractile vacuoles, one in the anterior and other one is present posteriorly. The function of contractile vacuoles is osmoregulation. That is, it regulates the water content of the body and may serve also in excretion of nitrogenous waste such as urea and ammonia. Excess of water within cytoplasm is secreted into the tubules of endoplasmic reticulum, and then the collected water goes through the nephridial tubules to the feeder canals, and the water is collected into ampulla of a series of 6 to 11 radiating canals that converge toward and discharge into each vacuoles. The contractile vacuoles contract alternatively at the intervals of 10 to 20 seconds. Reproduction in Paramecium chordatum. Paramecium chordatum reproduces asexually by transverse binary fission and also undergoes several types of nuclear reorganization such as conjugation, endomixis, autogamy, cytogamy, and hemixis. Transverse binary fission. Transverse binary fission is the commonest type of asexual reproduction in Paramecium. The plane of division is through the center of the cell and in a plane at right angles to the long axis of the body. Division of cell body as a whole is always preceded by division of nuclei. Indeed, it appears that reproduction is initiated by nuclear activity and division. Paramecium caudatum reproduces by transverse binary fission during favorable conditions. In binary fission, the micronucleus divides by mitosis into two daughter micronuclei, which moves to the opposite ends of the cell. The macronucleus elongates and divides transversely by amitosis. In meantime, a constriction furrow appears near the middle of the body and deepens until the cytoplasm is completely divided. The resulting two daughter paramecia are equal size, each containing a set of cell organelles. Of the two daughter paramecia produced, the anterior one is called protor and posterior one is called opisthe. The rate of multiplication depends upon external conditions such as food, temperature, age of the culture, population density, and the internal factors are heredity and physiology. Conjugation. Ordinarily, Paramecium caudatum multiplies by binary fission for long periods of time, but at intervals this may be interrupted by the joining of two animals along their oral surface for the sexual process of conjugation. The conjugation is defined as the temporary union of two individuals which mutually exchange micronuclear material. It is a unique type of sexual process in which two organisms separate soon after exchange of nuclear material. Each Paramecium caudatum species differs and exists in varieties or syngens. Further, within each syngen, there are number of mating types, usually two. The mating types remain morphologically identical, but they exhibit physiological differences. Process of conjugation. In conjugation, two Paramecium caudatum of opposite mating types of same variety come together with their ventral surfaces and unite by their oral grooves. Their cilia produce a substance on the surface of the body which causes adhesion of two conjugating paramecia. They stop feeding and their oral groove apparatus disappears. The pellicle and ectoplasm at the point of contact of both break down and a protoplasmic bridge is formed between two animals. Now, these individuals are called conjugants. In this condition, the conjugating pair swim actively and simultaneously a series of nuclear changes takes place. The macronucleus begins to disintegrate. The micronucleus of each conjugant divides twice, one of them being a reduction division. Thus, 4 haploid daughter micronuclei are produced in each conjugant. 3 of these 4 micronuclei disintegrate in each, so that only one remains. The remaining micronucleus of each conjugant divides mitotically into 2 unequal pronuclei, forming a larger stationary female pronucleus and a smaller active migratory male pronucleus. The migratory male pronucleus of one conjugant crosses over the protoplasmic bridge and fuses with the stationary pronucleus of other conjugant to form a synkaryon or conjugation nucleus, in which the diploid number of chromosomes is restored and there has been an exchange of hereditary material. The process has been compared with fertilization in higher animals, but this is not fertilization because no gametes are involved. The synkaryon of each X-conjugant divides 3 times to form 8 micronuclei in each X-conjugates. 4 of the 8 micronuclei enlarge and become macronuclei, and 3 of the 4 micronuclei disappears. The remaining micronucleus divides and at the same time the X conjugates divides by binary fission into two cells, each having two macronuclei and one micronucleus. The cells and their micronuclei divide a second time to form four paramecia from each X conjugate so that each has one macronucleus and one micronucleus. Significance of conjugation. A clone will die out if nuclear reorganization does not occur, but the clone can be rejuvenated to regain its former vigor by nuclear rearrangement. This nuclear reorganization is brought about by conjugation. Thus, conjugation is essential for continued binary fission. Autogamy of paramecium. Autogamy takes place in Paramecium aurelia, which has two micronuclei and one macronuclei. In the process of autogamy, the macronucleus breaks into DNA fragments and is absorbed into the cytoplasm. The two micronuclei divide by meiosis to form 8 daughter nuclei. Out of 8 daughter micronuclei, 7 disintegrates, remaining 1 micronucleus divides by mitosis to 2 gamete nuclei. The 2 gamete nuclei enter into the protoplasm for fusion. There they form zygote nucleus. The zygote nucleus divide twice to form 4 nuclei. Out of the 4 nuclei, 2 becomes micronuclei and 2 becomes macronuclei. The paramecium now divides and produces 2 other paramecia. Endomixis takes place in Paramecium aurelia. Paramecium aurelia has 1 macronucleus and 2 micronuclei. The macronucleus degenerates and disappears. The 2 micronuclei divides twice mitotically to form 8 daughter nuclei. Out of the 8 daughter nuclei, 6 micronuclei degenerates. Now, the paramecium divides into 2 daughter paramecium, each with 1 nucleus. Each micronucleus divides twice mitotically to form 4 daughter nuclei. Out of the 4 nucleus in each paramecium, 2 become macronuclei and 2 become micronuclei. Each micronuclei and the body of paramecium divides to produce 2 daughter paramecia. As a whole, endomixis of Paramecium aurelia yields 4 daughter nuclei from one single parent. Hemixis is primarily a process of macronuclear fragmentation and division without any unusual micronuclear activity. Diller classified hemixis into 4 types, namely A, B, C and D. But he also encountered all types in mass cultures of Paramecium caudatum and Paramecium multimicronucleatum. Type A is the simplest form of hemixis, characterized by a division of macronucleus into 2 or more parts. This division is not synchronized with micronuclear division. Type B is characterized by the extrusion of 1 to 2 or more chromatin balls from the macronucleus into the cytoplasm. Type C is characterized by simultaneous splitting of the macronucleus into 2 or more major portions and extrusion of macronuclear balls into cytoplasm. Type D is considered to represent pathologic conditions in which the macronucleus undergoes complete fragmentation into chromatin balls that eventually disappear from the cell. Micronuclei generally disappear before the dissolution of the macronuclei. Cytogamy. In 1940, Wichmann has reported another sexual process in Paramecium caudatum, which he termed it as cytogamy. In cytogamy, there is no nuclear exchange. In this process, 2 paramecia come together by their ventral surfaces, but the pellicle of the 2 individuals does not break down. The early nuclear divisions are also similar to those of conjugation. The micronucleus of each individual divides thrice to form 8 micronuclei, 6 of which disintegrate in each individual. The two remaining micronuclei fuse to form a synkaryon in each cell. The animals now separate. Cytogamy differs from autogamy in that there are two animals in contact with each other, but it resembles autogamy and conjugation in the fusion of the two pronuclei. Cytogamy differs from conjugation in that there is no nuclear exchange between the two animals which come together. The process is completed in about 13 hours. Disease-causing characters in Paramecium. Trypanosoma protozoa. Members of the genus Trypanosoma are flagellate protozoa that causes sleeping sickness, which is common in Africa. They also cause Chagas disease, which is common in South America. The parasites are spread by insect vectors. Trypanosoma parasites enter a person's blood, then the vector bites. Then they spread to other tissues and organs. The diseases may be fatal without medical treatment. Giardia protozoa. Giardia or flagellate protozoa that causes giardiasis. The parasites enter the body through food or water that has been contaminated by fecus of infected peoples or animals. The protozoa attach to the lining of the host's small intestine, where they prevent the host from fully absorbing nutrients. They may also cause diarrhea, abdominal pain, and fever. Plasmodium protozoa. Plasmodium protozoa causes malaria. The parasites are spread by a mosquito vector. Parasites enter a host's blood through the bite of an infected mosquito. The parasites infect the host's red blood cells, causing symptoms such as fever, joint pain, anemia, and fatigue. Malaria is common in tropical and subtropical climates throughout the world. In fact, malaria is one of the most common infectious diseases on the planet. Malaria is also a very serious disease. It kills several million people each year. Most of them are children. A vaccine to malaria is in a possibility. Ecological importance of phylum Protozoa. The protozoans are helpful in providing food to man, insects, fish, and other animals. They act as insect control. They help in sanitation by the purification of water. In the oil exploration, exact locations of oil deposits are identified with the help of these protozoans. The protozoans are used in scientific study in laboratories.